Hey everyone, Chris from ClaretBio here. Thanks so much for joining the webinar. Today I'm going to be talking about cell-free DNA methylation. I've got some history to share, I've got some data to show, and ultimately at the end of this video, I hope you get a better picture of why your choice in library prep chemistry really dictates the methylation results you see in your cell-free DNA methylation sequencing results. I hope you enjoy. Let's get started. So by the early 1900s, the four bases that made up DNA were known to the research community. Yet DNA still remained an enigma. It was, on one hand, it was clearly important to the cell because it was present, but on the other hand, it was largely repetitive and chemically inert. So researchers didn't attribute, at this point, DNA to having any broader biological function. That role was still attributed to proteins at the time. So fast forward through about half a century and 2 world wars, and researchers were getting back into studying the chemical constituents that make up DNA. And one of these researchers was Dr. Hotchkiss, who was an American biochemist based out of the Rockefeller Institute for Medical Research in New York. The way he was studying DNA was that he would take calf thymus DNA, he would hydrolyze it to separate it into its chemical constituents, and then he would run it out on paper chromatography. When he was doing this, he was expecting to see 4 spots on the paper chromatography that were corresponding to the 4 bases in DNA, A, C, G, and T. However, when he ran his experiment over and over, he kept seeing a 5th spot. Now, this 5th spot was directly above cytosine. So it was faint, it was reproducible, it was there, it couldn't be dismissed. So it was cytosine, but it wasn't cytosine. In fact, it was an epicytosine. And at this point, epi was just derived from the Latin term meaning above. So directly above cytosine, epicytosine. By 1949, Hotchkiss had chemically identified this spot as 5-methylcytosine. So on one hand, this was a profound result. DNA was no longer just 4 bases. It could have modifiers, it could have additions, it was mutable. On the other hand, researchers really didn't know what to make of this. Keep in mind, this was in the late 40s. This would be 4 years before Watson and Crick unveiled the structure of the double helix, many years before the idea of the genetic code even was proposed, and decades before the idea of epigenetics even came about. In fact, it really wasn't until the late 70s when 2 researchers working independently proposed the idea of epigenetics and gene regulation through this 5-methylcytosine marker. So these 2 researchers were Art Riggs, working out of the US, and Robin Holliday, working out of the UK. You may know Robin Holliday from another important discovery, that being the Holliday junction. But in the 70s, they proposed that 5-methylcytosine could be a regulatory mark that was on DNA, transferred through replication, replicated truly, but didn't really affect what DNA encoded for. Now, of course, through decades of research past that, their idea would largely be proven true. Not largely, it would be proven true. And 5-methylcytosine today is now known as the epigenetic mark that kind of controls gene silencing, chromatin structure and organization, what else? Heterochromatin, silenced region, open region, developmental kind of gene regulation in developing cells. And I know I'm skipping other stuff, but yeah. So that's the story of how 5-methylcytosine came out. Now, at the same time as Dr. Hotchkiss was studying and learning about 5-methylcytosine, there were 2 researchers working out of Paris. And these 2 researchers were Paul Mandel and Pierre Métis. And their job, not their job, but their research was to identify all the chemical constituents that made up blood. And they were doing this through techniques such as protein precipitation, UV absorbance measurements, acid extraction. And through their studies, they made an interesting discovery. They found that when you removed all the cells from blood plasma, nucleic acids still were present in the blood plasma. They wrote up their findings in the proceedings of their National Academy of Sciences journal, and it was just entitled Nucleic Acids in Human Blood Plasma. Similar to Dr. Hotchkiss and the discovery of 5-methylcytosine, the research community at large didn't know what to make of this discovery of cell-free DNA. It was an interesting observation, but it was largely thought to just be cellular debris. DNA in the blood, just cellular debris. So this observation lived in relative obscurity for several years, even decades. Studies in the 60s and 70s resurfaced the observation of cell-free DNA. Researchers would notice that patients with certain autoimmune diseases, such as lupus, had large amounts of DNA in their blood plasma. But patients with advanced stages of cancer would have large amounts of cell-free DNA floating in their blood plasma without cells present. But again, this was just largely dismissed as cellular debris. It really wasn't until the advent of PCR that the importance of cell-free DNA began taking shape. In the late 80s, researchers found SNP mutations, structural variants, single nucleotide variations associated with advanced cancers in the blood plasma of patients with said cancer. So at this point, late 80s, cell-free DNA is starting to be more popular. And then cell-free DNA really took hold in the late 90s with two kind of landmark studies. The first study being that researchers showed that fetal cell-free DNA, existed in the maternal blood plasma of mothers. And they did this in a really interesting way, but simple way, by just showing that the Y chromosome DNA sequences were from a male fetus were in the maternal blood plasma, linking the fetus cell-free DNA into the mom. And then the second study basically was taking shape to show that cell-free DNA wasn't just fragments from chromosomes. It wasn't large chromosomal fragments. They were actually short molecules that would later be tied to nucleosome protected DNA and thereby being short kind of mononucleosome length. So between these two studies, cell-free DNA was finally definitively tied to apoptosis and a biological mechanism for clearing DNA from the cells. And this led to the whole revolution that is cell-free DNA today. So to kind of tie these two separate observations together, both 5-methylcytosine and cell-free DNA were discovered in the late 40s. However, researchers had no clue what to make of either of these discoveries until the late 70s for 5-methylcytosine and even up to the late 90s for cell-free DNA. And because of this, and for good reason, the two observations have largely been studied separately. 5-methylcytosine as a regulatory marker in chromosomal and developmental cells, and cell-free DNA as apoptotic DNA that is shed from patients with tumor samples or pregnancy patients or transplant patients. And it really hasn't been until recently that we've been able to study the two together. And it turns out that when you study methylation and cell-free DNA, the results are just much more fascinating and make cell-free DNA even more interesting than it is, basically. So with that, let's get into the data. Now, methylation is not distributed randomly across the genome, as it regulates, expression in differentiated cells, active regions, silent regions, heterochromatin, all that good stuff. However, when we look at the methylation signature of bulk genomic DNA, what we're essentially seeing is just an average across many chromatin states and often many cell types. And because bulk genomic DNA kind of samples the genome indiscriminately, highly methylated regions and unmethylated regulatory regions are all mixed together. And that's why you get kind of a flattening of the tissue-specific methylation curve, and you just kind of get 50% methylation at CpG sites. One cool aspect of cell-free DNA methylation is that it is non-random. Cell-free DNA is fundamentally different than genomic DNA because it's not a random sampling of the genome. It's selected. Extracted cell-free DNA molecules are essentially the survivor molecules of apoptosis and in some cases necrosis, where nucleases destroy any and all DNA they can, and what survives in the bloodstream is methyl-enriched for specific regulatory states, and these are mostly closed chromatin and transcriptionally inactive regions. So it's kind of a self-purifying selection because methylated DNA is not expressed, it's closed, it's wrapped around nucleosomes, it's protected from nucleases, and so what you see is highly methylated DNA in the cell-free DNA. So it's biased towards the regulatory landscape of the cells that release them. Now, up until this point in our data section here, we've been talking about the methylation of cell-free DNA molecules as if the methylation state of the molecule that we're sequencing is an intrinsic property of the molecule itself. But I know it's complicated, but there's an assumption built in there. And the assumption is that what we sequence is what is the native methylation state of the molecule. And that's not entirely true at all, because what we sequence depends on the chemistry that gets us to the sequencing data. And for that to make sense, we need to talk about how library molecules are prepared for DNA sequencing, and specifically end repair itself. When we work with genomic DNA, we usually start with these long intact molecules, and we mechanically or we enzymatically shear them. And so this process just so happens to tend to produce relatively clean ends. They're mostly blunt with very short overhangs, as I'm kind of detailing here. And so that's what you see with long DNA, whether it be genomic DNA or DNA derived from somewhat degraded samples such as FFPE DNA. But cell-free DNA, on the other hand, is completely different. It's generated biologically, not mechanically, and nucleases progressively trim off DNA from nucleosomes, and this produces naturally short fragments that are about 167 base pairs in length, but also fragments that routinely have uneven and recessed ends, as I'm kind of detailing here. More recessed ends on either the 5' or 3' end. So most sequencing workflows, most NGS library prep sequencing workflows that involve a ligation method, they include an end repair step. And the goal here is simple. Just convert all fragments into clean, blunt molecules such that Taq polymerase can add its A tail and the adapters can ligate efficiently through a TA ligation strategy. It's a great strategy, and it works well for almost all types of DNA inputs. However, not so much for cell-free DNA, because end repair doesn't just fix the DNA ends, it rewrites them in a way. So let me kind of show this easy example here. So here you see a DNA fragment with a recessed 3' end or a 5' overhang. And then you have the methylation status of this molecule in these filled in lollipops or empty lollipops on top of the DNA. So if a methylated cytosine happens to fall on a recessed 3' end repair will fill in that position with a newly synthesized cytosine. But it's not methylation aware. So the cytosine it just fills in is unmethylated. And biologically speaking, that shouldn't happen. I mean, it does happen when replication in the cells take place. There is a hemimethylated state in the newly synthesized strand, but then enzymes such as DMNT1 and its like enzymes come in and they recognize that the strand that the old strand is methylated, so it turns this into a methylated cytosine. And that just simply doesn't happen here. So with that in mind, let's go back to our methylation signature plots and fill in the whole story, because I kind of left some information out the first time around. So in genomic DNA, this end repair induced methylation loss does indeed happen, as you can see kind of the drop off of methylation in the double-stranded prep with end repair here. But because the overhangs produced by mechanical or enzymatic shearing methods are short, this effect is really confined to the very end of the read, the last 2 to 5 base pairs. And this is nothing that trimming a few bases off the end of the read informatically after sequencing, after the entire prep, won't fix. However, in cell-free DNA, these recessed ends are longer and the overall methylation is higher, as I showed in that original plot. So the effect is just magnified more so. So what you should notice here is not just that cell-free DNA looks different from genomic DNA. We established that in the earlier plot, higher overall methylation. But what you should notice is that employing a library prep that utilizes end repair with cell-free DNA causes a drop in the observed methylation level starting as early as 50 base pairs into the molecule and then just dropping off precipitously in the last 30 base pairs or so, indicating that the majority, or not the majority, but when there is a terminal overhang or recessed fragment in cell-free DNA, it's mostly in this in the range of 10 to 30 base pairs of overhang, but some molecules can be longer. And whether this is an overhang or a nick in the cell-free DNA is important biologically speaking, but for the process of library prep molecules, it essentially means the same thing. So to understand kind of what this means for the data, it helps to look at methylation not as a position along the read, which is what we've been looking at thus far, but how this changes in terms of distribution across CpG sites. In other words, when we prepare cell-free DNA libraries with and without end repair, how does the overall distribution of CpG methylation change? So this figure here shows the difference in density between cell-free DNA libraries prepared with single-stranded library prep that doesn't have end repair and a double-stranded library prep that does have an end repair. And the plot is set at 0, where anything where you see a difference in the single-stranded prep is shown in positive numbers on the y-axis, and any CpG methylation state that is methylation percent basically at a CpG loci is for double-stranded prep is seen, in the negative numbers here. So a position at 0 essentially means there is no difference between the double-stranded prep and the single-stranded prep. So to understand this figure, it really helps to kind of just think about cell-free DNA molecules in a single cell state. In a single cell state, per CpG loci, a CpG is either going to be not methylated or fully methylated. And that just depends on the differentiation of the cell and what, the function of the cell is. Cell-free DNA is a little bit different because the DNA fragments are coming from all sorts of different cells. So the CpG at the loci, per fragment, it's going to either be methylated or not. But at the loci in the genome, you got many different reads covering that, so it can be, partially methylated. However, the effect of that is largely normalized out by the single-stranded prep, because that is sampling the cell-free DNA without end repair. So whatever you see here in the kind of hemi-methylated states is just a result of end repair. And the fact that you have more hemi-methylated or more partial CpG methylation states closer to 100% is just indicative of this end repair process. Overall, this plot supports the initial plots, but it is a nice way to kind of visualize this overall, that, there are just more end repair based hypomethylation states at the end of molecules, and that's contributing to this. So this is interesting kind of results, and it's very cool to see that cell-free DNA fragments are, end distinct from sheared longer DNAs, and this end difference is due purely to the biology of how these cell-free DNA molecules are produced in the cell and then in in the blood plasma itself. So that in itself is very interesting, and it's a cool fragmentomics observation, but how does this kind of affect translational implications? How does this affect how we study cell-free DNA for its liquid biopsy purposes? And so for that, we need to talk a little bit about tissue of origin studies. So different cells have different methylation patterns, and you can take cells from different tissues, and you can do this in either a single cell or kind of a bulk format, as long as you FACS sort the cells correctly and you have the right markers, so you assume you have a homogeneous population. And remember, you're taking the DNA from cells, so even if you're shearing them, the ends terminize, you can kind of get rid of that because it's only the last 2 to 5 base pairs, so it doesn't matter. But you can do all of this exhaustive studies, and you can get a catalog of methylation patterns for these different tissue types, which is, what I'm showing here in kind of this encyclopedia, this old book here. And from now on, I'll be talking about it as an atlas of kind of methylation patterns. So using this atlas, this catalog, this encyclopedia of methylation signatures from certain tissues and cells of origin, you can now take a cell-free DNA fragment and you can compare the methylation pattern at that cell-free DNA fragment level, which is about that 167 bases, and you can kind of attribute that to the tissue of origin. So if a fragment has got, a certain pattern, methylation pattern at a certain loci, it can come from, the blood cells itself, or it can come from, a more or a less prevalent cell type, and you can in the cell-free DNA, and you can understand that through the fragmentation pattern. So for the next figure that I'm going to show you, we used an atlas from the Kaplan lab, which is a lab out of the Hebrew University of Jerusalem. Largely they're a computational lab, but they develop large methylation atlas cell sets, specifically, for other reasons, but also so that researchers can look at cell-free DNA tissues of origin. So with that, let's kind of dive into this figure here, which is the crux of the whole research, what we've built up to at this point. So with this context now about tissue of origin, we can ask a very focused question here. Does end repair change how well cell-free DNA methylation data can be matched back to tissue methylation signatures? So in this figure, the x-axis shows your R-square value. So you could think of this as asking, if I take the best possible weighted combination of tissue profiles from the atlas, how much variation in the cell-free DNA methylation data can I account for here? An R-squared value of 1 means a perfect explanation. An R-squared value closer to 0 would mean the atlas is doing a poor job of explaining the data that we have in hand. So in this context, you can really think of R-squared as acting as a proxy for how much of the original tissue-specific methylation information survives in the library prep process. So this is a ridge plot, and the ridges represent a distribution of these R-square values across repeated train test iterations for the same cell-free DNA extracts. So these ridges capture both performance and stability across something like 100 individual train test runs that we ran. And the rows respond to different CpG site methylation cutoffs, such as only taking reads that have less than 0.2 CpGs methylated or greater than 0.8, 0.1, 0.9. And I'll get to the kind of control as a second. And the reason we do this is because CpG values near 0 or near fully methylated tend to carry the strongest tissue-specific signal. So as a control here, we also included a set of CpG sites that are consistently unmethylated, but still tissue informative. This control is therefore an insensitive to end repair methylation loss control that we can look at. So what you can see, what emerges here is a consistent pattern for CpG subsets that include the highly methylated sites. And it shows that libraries that preserve the native cell-free DNA termini show higher R-squared values, whereas libraries prepared with end repair show reduced concordance. Importantly, this difference largely disappears in the unmethylated control set, indicating that the performance differences that we see are concentrated in these kind of methylated regions. So in other words, to sum this whole distribution up, the distribution of methylation profiles that we saw earlier doesn't just change the shape of the data, it directly affects how well the data interprets cell-free DNA biology. So the main point of this talk is hopefully kind of simple and straightforward. I did show some history at the beginning, which I think is fascinating to know kind of where the word epigenetics originated from, and that it was from this Roland Hotchkiss experiments in the late 1940s about epicytosine just being slightly higher above cytosine on paper chromatography. Is just fascinating. But history, and then, just also the discoveries of cell-free DNA and epigenetics, and their discoveries largely before they could even be explained or the tools were in place to kind of fully understand what they were. Super fascinating. But the data here is meant to show something that's cool, but hopefully straightforward, right? That cell-free DNA is just biologically produced. Because it's biologically produced, it has different ends than genomically sheared DNA. These ends, not only the ends, but the methylation status of cell-free DNA is kind of a purifying selection for more methylated DNA in general. So there's kind of this perfect storm, right? Where cell-free DNA is highly methylated, but it also has a lot of overhangs. And so when you have end repair added into this equation, you erase a lot of the methylation signature. So very interesting, and really just leaves you with one kind of question here. Which library prep will you choose? All right, thanks everybody. That was my attempt at kind of a higher level production of this webinar, since afterwards it's going to be put on YouTube, and you can try to have more on YouTube. I like to have something that's a little bit more engaging. So this was my first try at that. I do want to mention a few things here before I kind of go live, because spoiler alert, this was recorded earlier. But the things I want to mention are the credits. First of all, this observation of these long overhangs in cell-free DNA using methylation was not discovered by us. This comes out of Dennis Lowe's lab. Dennis Lowe actually being the researcher in the intro who I talked about, who discovered fetal cell-free DNA in maternal blood plasma, leading to the whole NIPT kind of analyses and diagnoses and all the cool stuff we can do with cell-free DNA in neonatal babies. But anyways, check out this paper. Very cool. They don't kind of look at tissue of origin here. They use the methylation signature and end repair to kind of examine how long overhangs are in double-stranded DNA. But very awesome stuff. Again, not our initial observation. The second paper is kind of this landmark paper that comes out of Jay Shinjuri's lab at University of Washington. And I mentioned them because I took one of their figures to show kind of the nuclease, the DNA wrapped around the nucleosome. So, but also very cool paper, landmark paper out of Cell. Go check it out. And then I will plug our paper, which goes into way more detail than I did here with how end repair affects tissue of origin, shows duplicate cell-free DNA samples, some more results about the tissue of origin, and yeah, so check out our paper in the Journal of Liquid Biopsy, and then of course our kind of initial paper, which is seriously our Claret single-stranded library prep that is used for cell-free DNA analysis. So I'll put this kind of as the full screen credit. And with that, thanks for watching. I'm happy to take questions while I turn it back to live Chris now. So thank you.